You are Mr. 1-8. Niranjan Ghoshal. Okay, you are one of them. Poster number 17. Shundavi Ghosh. You are another one. And another one is poster number 9. Oindrila Acharya. Okay. All you have to do, the three of you, we have to, in the next session, session 3, as you note in your program, that you have to give a 5-minute talk on your work. I believe you received an earlier notification that in the event of your being selected for the poster award, you have to give and work out and deliver a talk of 5 minutes in 3 slides, not more than that. I'm sure you are all prepared for it. So these persons, congratulations to them. Poster 18, poster 17, and poster 9. All 3 of you will have to deliver the talks of 5 minutes after this session, in the 3rd session. We begin the 2nd session now, and I would, this is the Uma Ganguly Memorial Scientific Session by invited speakers. And I'd like to invite Professor Sharmila Sengupta and Professor Subrata Majumdar to chair this session. For those who we are not familiar with the contributions of Dr. Sharmila Sengupta and Professor Subrata Majumdar, both of them are well known to the community as cancer specialists. Incidentally, Dr. Sharmila Sengupta at one time ran the NIBMG, National Institute of Biomedical Genomics, as director in charge for more than a couple of years, I believe, after Professor Parthapratim Majumdar retired. And the second, not really, is 1 and 2 doesn't mean anything. And Professor Subrata Majumdar, known to me for a longer time at the Bose Institute, is also another cancer specialist, and he is now an ICMR Emeritus Scientist of Bose Institute. 2 of them will chair the session. May I request these 2 persons to kindly conduct the session of 3 lectures by another NIBMG scientist first, Arindam, and then Pritha Roy and Shiv Shankar Roy. And again, I have personal interaction with all the 3 speakers, particularly Pritha and Shiv Shankar, who happen to be the students of Vishwavarati Life Sciences. And okay, let me stop right there. May I invite Sharmila and Subrata to kindly conduct the session.
Hello, thank you very much, Arunda and Oshimadi, for inviting me as a judge for the poster sessions, as well as a chairperson of this seminar. Umadi was very close to me actually when she was at NICED. I knew her for a long time, but she used to come to my lab actually when she was coming to meet Porimalda at Bose Institute, and she used to visit my lab. And she was very nice actually, and used to work in the signal transduction mechanisms. And she tried to collaborate with me, but I didn't get any time to collaborate with her. But anyway, she was very good. So without taking much time, I'd like to request Dr. Arindam Moitro, or Professor Arindam Moitro, the Associate Director and Professor of BRIC, National Institute of Biomedical Genomics, where I was involved in this from the scratch actually. when the national brick NIBMG was not there. So I was there from the ground level, but now I'm retired from anywhere from the scientific society. So Arindam was the temporary, he was director for a few months over there. And he did very nice work, published huge number of papers, and most important is the Nature Communications, Cancer Science, Genomics, Lancet. So published so many papers, and he is from the chemistry and from Presidency College, ex-student of the biochemistry department, master degree where we are from, and he did PhD from AIMS, New Delhi. So Arindam is received the Fellow of West Bengal Academy of Science and Technology Award, and he is involved in technical committee of many departments, many institutes, including a scientific advisory committee of ICMR, Regional Medical Research Center, Northeast region. So I would like to request Dr. or Professor Arindam Moitra to come up on the stage to deliver your talk. Where is your talk? I don't know, I can see it. So you can read your title, and then you start your talk. Thank you very much. Oh, your 30 minutes talk, 30 minutes.
Thank you. Thank you, Shubhro Dada. I mean, to you it's just Arindam. Thank you very much, sir and ma'am, for inviting me and many others who are part of this. It's always a pleasure to come to SNU for various reasons. We get many occasions to come to SNU, and we always ensure that we do not miss them. And in particular, this particular event is first time I am coming, and I learned about it, and I understand it's a very special event, and my respects to the entire society which has invited me. So I will be continuing our discussions on cancer. I will be talking to you about understanding the oral cancer tumor ecosystem. So in the morning, Dipoman made many statements which are a kind of leading to my talk. One of them was, he said that the cancer tumor is actually a whole bunch of cells, and there are many different kind of cells. So my talk will actually show you a genomic definition of all these whole bunch of cells and different kinds of cells in the tumor ecosystem. But first things first, there are a few people who actually did the work. And I was probably, I worked with them, and I'm a facilitator. The first and foremost was Dr. Siladin Kurkalang. She did her PhD from Nehu and joined as postdoc. Right now she is in University of Chicago, but she was very much key person who initiated this actual single cell RNA sequencing method that I am showing it to you on oral cancer. The other two are my PhD students. Shumitabo has just finished his PhD. He was a primary goer in this, and he is writing his thesis. And Orunima is right now in a third year of PhD and carrying forward the baton, doing a deeper work on this tumor ecosystem, understanding of tumor ecosystem. There are few postdocs. Shomnath is from ISCB, did his PhD from ISCB, from Udayada's lab, had been with us for a brief while, and he played a very important role. Sanjoita is from IPGMER, did her PhD, and right now spearheading our venturing into from genomics to cancer biology. And last but not the least, there are two young people I should mention who spent some time with us, one previous year, one this year. Shubhashree is from doing her MS, was doing her MS in bioinformatics from Central University of South Bihar. And Orgho is actually a student of SNU. This year he spent 6 months with us in participating in this kind of work, and we are very impressed with him. So I'll be talking about our journey into this oral tumor ecosystem and what kind of insights we learned. Before that, we had, of course, many of you might know that we had been working on oral cancer, understanding the oral cancer in India, its genomic basis, what are the alterations in the genome which drive oral cancer in India. We also tried to understand what are the transcriptomic alterations in oral cancers, and we also found very interesting things related to the mitochondrial genome. Not going into those are from the past. But all these studies were done on biopsies of tumors. So what we did was we took the tumor biopsy, extracted the DNA or RNA, and then we did our analysis like whole genome sequencing, RNA sequencing, DNA methylation analysis, and so forth. In the process, what we do is I don't know, this doesn't work. Okay. This will work. This will work. So what we did was, we took this, when we did this, we extracted the DNA and RNA from a whole bunch of cells in the tumor. And these cells are varied kinds. They are malignant cells, they are non-malignant cells, different cell types. And we extracted the total analyte and we sequenced them. In the process, we had an average. I can give you an example that if we started singing in a song in a chorus in this hall, all we'll hear is the entire chorus, right? We will not be able to differentiate very good singers like you or very bad singers like me. But if there was some ability by which we could barcode everybody's voice, such that from the chorus, using the barcode, we can separate out each singer's performance from the chorus, then it would have been lovely. We could have resolved how each and every one of us sang. And exactly, the biology in most of these kind of situations is music of life. It's sung in a chorus, but there are individual components which ultimately matter how the chorus performs. So similarly, we understood that there are many things here going on which we are not able to catch. And that's because we are looking at a chorus of the sequencing data that we are looking at. On the other hand, if we could actually differentiate them into various cells, so let us take for example the transcriptome data. Gene expression data, if we could collect and then identify where each of the cells, what was the gene expression patterns obtained, that would have given us a lot of information. And that's actually what helps us to differentiate between the healthy and the pathological state. And oral cancer is an example of that. So this led to the advent of these technologies like the single-cell RNA sequencing. Sushantada mentioned about human cell atlas, so we participated in human cell atlas. And single-cell RNA sequencing and these kind of technologies were fueled the formation and the activities of human cell atlas, where we are able to sequence each cell, in a figurative sense, and identify each cell's gene expression. We saw that the tumors are a complex ecosystems, and this is a perfect challenge for this kind of single-cell analysis to be done. We understand that oral cancer has a lot of heterogeneity of clinical outcomes. I'm not going into details of that, but this is where we can apply. If we could make out how each of the cells in the tumor ecosystem express different or 20,000 odd genes, we might get some clue into how they are performing. So what we did was initially difficult to convince clinicians. We managed to talk to a very exciting clinician who could excite in the Dr. R. Ahmed Dental College and Hospital. In his OPDs, we collected biopsies from patients who were suspected and then finally diagnosed to have oral cancer. We collected those tissues in 2 hours' time. Initially, when we are developing protocols, we had a window time of 2 hours to bring the tissue back to the lab from the clinic after isolation and process. Nowadays, we have got over that challenge using the suburban railway service from Sialda to Kalyani, and processed them into the lab, or worked overnight in the lab and generated the data. So we did single-cell RNA sequencing of treatment-naive, freshly resected treatment-naive oral cancer tumors, and we analyzed the data using this kind of plots. These are known as UMAP, where each dot represents a cell, and The two dots are close or far apart depending on the expression similarity or difference of the 20,000 genes. So this is a dimensionality reduction technique. So if I look at, so these, and then looking at what are the genes that are expressed by each of these clusters, we can actually identify what cell types are there. So we can identify endothelial cells, epithelial cells, myeloid cells, mast cells, fibroblasts, and so forth. So this is what we did for all these initial batch of 12 patients, and we also developed methods by which from epithelial cells we can actually classify what are malignant cells and what are non-malignant cells. Remember, there are no cellular markers to identify malignant cells per se. So we had to use different methods by which we could assess based on expression levels and estimated copy number analysis, not going into that. What we found immediately was very interesting data, which we never got from bulk. When we looked at non-malignant cells, so if you see this, these are clusters of all non-malignant cells, which we annotated using the expression of the known marker genes. The non-malignant cells all clustered by the cell type. That means all myeloid cells clustered together, all fibroblasts clustered together, all T cells clustered together, and so forth. So they do not cluster by anything else. No matter from which patient the cells have been obtained, they will express similarly. When we move to malignant cells, we found that there is a big change. So there are some clusters which are specific to patients. So some of the malignant cells are actually specific for each tumor. And between two patients, they have a wide difference in gene expression pattern. So that means there is a lot more gene expression heterogeneity in malignant cells compared to non-malignant cells. We also moved into, so we looked into the various other kinds like non-malignant cells. We wanted to identify what are the different non-malignant cells, and we found that there are various subsets of non-malignant cells like CD4, CD8 cells. We also identified a very interesting set which we called as double negative T cells. That means they do not have expression of CD4, they do not have expression of CD8, and they're T cells, CD3+. So we did something else. Because we have large number of cells with different kind of expressions, we can recapitulate theoretically a trajectory, like evolutionary trajectory called in pseudotime. We call it pseudotime analysis. So we can see that these are something like that, and we found that there is a separation of double negative T cells from the CD4 or CD8 cells, and we found that there is an expression of a gene called PLCG2 in many of these double negative T cells. PLCG2 is phospholipase C gamma 2. It's a pro-inflammatory protein. So we also found that there are macrophages. Very interesting data was that we found that TIP1 again talked about M1 and M2 in the morning. M1 are pro-inflammatory, M2 are anti-inflammatory. So we found because OSCC are supposedly hot tumors, there are a lot of macrophages, and there is the M1 and M2, but we found there is a large chunk of cells which are actually showing expression of genes of M1 and M2. That means if you look at trajectory analysis, they are actually evolving. So what we suspect is they're evolving from M1 to M2. So they behave, we call it the M1, M2 phenotype. So it's in transition from pro-inflammatory. The macrophages are slowly turning out to be anti-inflammatory. This is for the benefit of the tumor cells to survive and thrive in the tumor ecosystem. Malignant cells we found belongs to two large groups. Interestingly again, so this particular group we found mostly was contributed from two patients. And what were the two patients' commonality? We found that they also have another additional feature. They all have something known as oral submucous fibrosis, OSMF. OSMF is very common in India. It is caused by chewing of areca nuts, supposedly. There's an extensive fibrosis happens, very painful, and these two patients, OSCC developed in the backdrop of OSMF, whereas in the other patients they do not. We found that there are three different malignant cell clusters. One group expressed immunoglobulin genes. and cancer testis genes. These are malignant cells, remember, epithelial malignant cells. The second group expressed something known as, these are like fetal-like epithelial gene signatures. So these are some of the genes being expressed by these malignant cells are those that are seen in the fetus. So this is something known as, now we call it as oncofetal reprogramming. So some of these cells are going back in time to de-differentiate, and that is what we are seeing. And the third group expressed, again a transition. We heard of EMT, epithelial to mesenchymal transition. Here they are expressing partial EMT. That means they are moving from epithelial to mesenchymal. It's in the midway, it is called partial EMT. So this was published in the Cancer Science paper last year, and then we have moved on from this study. We are now focusing on a few questions like oncofetal reprogramming, double negative T cells, the M1, M2 phenotype, and there's something else also that I will not talk about. So OSMF, as I told you, is a very known problem. It's extensive in India, and there are issues. So we did some more studies with more patients of OSMF associated OSCC and OSCC patients, and we found that there are 4 clusters. So with a larger number of patients, we are seeing 4 clusters of cells, malignant cells, which are divided into 2 groups. And this group belongs to patients, cells from patients who have OSMF, whereas this group belongs to patients mostly who have only OSCC, not an OSMF background. And these are some kind of analysis where we found that these clusters of cells have enrichments which are different in OSMF associated OSCC versus OSCC. We found that cluster 1 and 3, which are primarily present in OSMF, which are primarily present in patients who do not have OSMF, have genes which express energy production and stress adaptation programs, and also some amount of epithelial and immune defense. So these are more aggressive phenotype of malignant cells. Whereas patients who, so clusters 2 and 4 come from patients who have OSMF, concomitant OSMF. And these 2 and 4 express programs which are like cell communication and structure, they are immune signaling and cell death. So we can get a sense that OSCC associated with OSMF probably has a better immune response in the tumor microenvironment, and hence might have a better prognosis. So we then looked into and found that, again, cutting a long story short, that I talked about the genes which cells which are expressing fetal genes. There are 2 types. One which are more like present in OSCC only patients, and they are more kind of, so this branch 4, and they have very aggressive cell state, whereas the other one which is present in mostly patients with OSMF and OSCC, they have more enriched with immune modulatory functions. So this is going into real deep detail. With double negative T cells, again we found that the double negative T cells actually branch out into 2 different forms. One where double negative T cells which express PLCG2, that phospholipase C gamma 2, that gene, whereas the other group of double negative cells do not express PLCG2. Now, this is very important because we are now homing in on this PLCG2. We suspect that these T cells are actually CD8 cells which have lost their CD8 expression when they moved into the tumor microenvironment. And in response to high calcium, there is an increase in PLCG2 expression in these cells, and they are actually moving into, basically, they are T cells which are moving into a quiescent state, because we saw that when this PLCG2 high cells also have a gene which is highly expressed, and this is correlated with the progression. It is called BTG2. Now, BTG2 has been shown to be associated with T cell quiescence. So basically, PLCG2 high double negative T cells in the OSCC microenvironment is actually showing that T cells are entering. They are hot tumors, but the T cells are becoming quiescent. So ultimately, there is a loss of immune response to the tumor. So this is like we are moving into slowly from a genomics lab to understanding how does the biology pan out. Many people asked us, do they really exist, or they are figments of your data analysis? So we have started doing FACS, and we have done ICC, and we have shown that these PLCG2 positive double negative T cells are more enriched in tumors of patients compared to healthy, those we isolate from healthy individuals' blood. Finally, we also looked into the malignant cells, and we found that there is, I talked about this M1, M2 dual phenotype, that also exists. So this is a plot where on the right side are OSMF associated OSCC, on the left side is OSCC, and we found that there is two kinds of M1, M2 dual phenotype macrophages. So when we looked into deeper detail, we found that they are actually M1, M2 cells which are more like M1, that means dominated by M1, but still they are M1, M2. And then there are cells which are M1, M2, which are more like M2. So these are like working out the details of the progression from M1 to M2. So macrophages initially in the OSCC tumor microenvironment are M1, but then over time they are becoming from pro-inflammatory to anti-inflammatory, and hence again helping the tumor to grow. So basically I will cut the story here. We have mapped extensive transcriptional diversity at cellular level. We have found that OSCC associated, OSMF associated OSCC tumors actually have different kind of expression profiles, and there is a dominance of, in malignant cells, there is a dominance of expression of programs like partial EMT, oncofetal reprogramming, and immunoglobulin kappa chain gene expressions. We have also found that there is a difference in T cell immune expression programs. So the OSMF associated OSCC show more cell death associated immune functions compared to OSCC. And we found that there are distinct multiple states of oncofetally reprogrammed malignant cells. There are cells like DNT where we found that there are two different phenotypes, one expressing PLCG2 gene, and which might be associated with the quiescence of the T cells. So we are actually taking this up. We are exploring now how important will this be for an immunotherapy, in terms of immunotherapy, whether we can modify expression of these double negative PLCG2 plus T cells so that we can downregulate their PLCG2 and use that to treat OSM patients. Don't know. I mean, it is easier said than done, and it's very easy for me to say that because I am the least knowledgeable in this matter, but a lot of excited about it, and it seems that the excitement is infectious. And then we are also seeing these oncofetally reprogrammed cells. We have done the analysis on malignant cells, and we find that there are different kinds of oncofetally reprogrammed malignant cells which might have a very important role in tumorigenesis and progression. We are now trying to look at the whole lot of the non-malignant cells which might also be cross-talking with these oncofetally reprogrammed cells and understand what kind of program changes in the gene expression they might be having, and that might be affecting the tumor ecosystem. So I think I'm done with time. I did well with time. So I will end here. My email address, and there are other programs that we are doing, particularly in single-cell transcriptomics. We are working on placenta in preterm birth. We are also participating in a study, international study called the Asian Immune Diversity Atlas, where we are doing single-cell transcriptomics of healthy peripheral blood cells of healthy Indian individuals. It's part of a larger Asian initiative, and we have very interesting questions there as well we are asking. And finally, please follow us on the social media in NIBMG. I'm done.
Thank you very much. You are in time, or maybe 10 minutes ahead, before actually. So any question, please? Shamalda first.
So this is actually the question that we are also asking. So when we found these DNT cells, we initially thought that they are more kind of evolving, but then we talked with many other cancer biologists, and what we understand is more feasible is they are CD8 plus cells which have moved in, And there are signals from, now we are looking at, because of the single cell data, we can also look at cell-cell interactions. So we are trying to map the cell-cell interactions. We feel that there are malignant cells are giving them specific signals. So they are losing their CD8 expression, and they are starting to express PLCG2.
So signal means, because for T cell stimulation, one would expect that it should be antigen specific expansion where the co-receptors are needed. So here co-receptors are missing, only probably they are having TCR, and that may not be adequate for their expansion. So what are the signal actually that is driving this population?
That will map out, but what important thing is they are probably not expanding there. So whatever has moved in, they are getting into a quiescent state.
So what is their frequency?
The T cells are a lot. I mean,
DN cells are quite frequent.
So let me take you back to this. So if you look at this, just one second. So you can see that these are the double negative T cells in this whole bunch of T cells. And if you look at the entire tumor ecosystem, so that these are the whole bunch of T cells.
So the B cells that you are showing, whether they are B10 phenotype?
We haven't looked in detail in B cells. We got caught up in the T cells. So, but definitely, overall the numbers of B cells are less. But
it could be less, but the B1 SL population, they are high producer of IL-10 and
we'll
TGF.
check that out. We'll check that out.
So that's the thing. Okay.
That will be a good thing to check out. Thank you.
Thank you.
Thank you. Please.
Excellent talk. So I have a question that may not be related to what you have been doing lately, but I think you are in a good position to address this. So one thing comes to my mind. Hello. So given the potential connection between microbiome and cancer, is a much talked about. So have you ever thought about just establishing some connection between oral cancer and oral microbiome dysregulation?
So from the single cell data, actually we can map, hypothetically, we can map cells which are in, so like whether they are HPV positive or negative. That's one question, because in OSCC now HPV is something that people look into. But like our bulk sequencing data, we didn't find any expression of HPV genes in associated genes in this particular patients that we looked at. But there are other, you are talking, so that takes care of the virome. But when you take up the microbiome, yes, we want to do it because we are capturing a lot of the data. And sometimes what can happen is looking at the expression of the genes of the human cells, we can actually understand if there is a signature for infection or not. It takes a lot of time. I want to do it, but someday probably I will do it. That's all I can say.
Please.
This may be a very, what I call very much non-relevant, because I am from social sciences. But since cancer affects both the social sciences, or does not make any differentiation while it affects. In case of tumor, malignant cells are found out, and you have talked about cell-to-cell interaction. Is there a possibility that the cancer biologist can suggest some preventive kind of course of action, or in a kind of awareness generation through preventive side of interaction? After all, this is for human beings. If they do not know, there will be no use of further research.
No, absolutely correct, ma'am. So what our bulk genomic data have shown, when we did a substantial number of whole genome sequencing of these oral tumors, we found telltale signatures of tobacco. So in the somatic mutation spectrum, you can identify which kind of somatic mutations are associated with what kind of mutagens. And tobacco has an established signature. And although all our patients are, in India we say that oral cancer is primarily caused by smokeless tobacco, which is like chewing of tobacco. So even in these, we could detect telltale evidences of tobacco. So we can say that, please don't use tobacco. Oral cancer is a direct fallout. Those mutations wouldn't have happened probably had tobacco not been used locally in the mouth. So that's a direct one. Regarding prevention, what we are trying to do is, there are two aspects. So one is, of course, from our genome sequence data, we have come up with now trying to form a panel of genes which can be used to screen people who have a pre-malignant lesion like oral submucous fibrosis or leukoplakia or other kind, and from a brushing of the mouth, that's where we want it to go. It's nowhere right now. But by sequencing those genes, we can say whether there are any of these mutations which are associated with oral cancer we are started to see in these people or not. Then we can alert them that please don't go any further. You are already crossed the nature's threshold. And the second point is that, like using these, like what we want to do, these double negative T cells, if you can take them forward, whether that can be used to, how do I say, treat pre-malignant lesions which are at a high risk of developing tumor. That can also prevent people from getting into a frank oral cancer. So these are what we can do.
Thank you very much.
Rita.
Where you have seen this DNT population, what is the clinical outcome?
I wish we could. So when 2019 I started talking to clinicians, people who treat cancer, and told that I will use these kind of studies to understand oral cancer, clinicians didn't believe me. They said that nothing can happen out of this. ये तो states में होता है, यहाँ पे कुछ नहीं होने वाला है. So I could not find anybody who would be willing to contribute patients. So finally I got hold of a pathologist who was very excited in Dr. R. Ahmed Dental College. Dental College and hospital, there will be no follow-up. They will diagnose the patient with cancer, and then in most cases in R. Ahmed, what happens, I understand, is again a social tragedy, because the patients from there go back to their home in deep rural part, wherever they come from, and they are left to die.
So, any questions from the students? No.
Add to that, what we are doing is now a lot of people are showing interest, and we are planning to coordinate with clinicians to look at follow-up data. And we have excellent follow-up data with our bulk tube, the data we generated on the bulk samples because of ICGC and all. Now what we are trying to develop are methods by which using these single cell signatures, we can track the bulk data and then see how the follow-up works out.
Students, any questions? I have small questions. The double negative T cells or DNT cells produce enhanced PLC gamma 2 expressions more. It is PLC gamma 2A or B? You don't know that.
PLCG 2A.
2A.
Probably.
Okay. So have you checked any ideas about the downstream signaling? Because in PLC gamma 2 downstream, lots of phospholipase protein kinase C alpha is expressed or more expressed in cancer cells. Do you think that is any relations with your
No, that's what we want to do. That's what we want to do now. Okay. I don't want to do any more single cell RNA sequencing. I just want to pick up these leads and I want to drive forward. So PLC gamma 2 and double negative T cells is right there up on my list.
But the downstream signal is very important because in feedback mechanisms, that if you can inhibit some of the parameters downstream, then it can can block the PLC gamma 2
from the tumor,
also.
we are trying to isolate now the DNT cells. These kind of sort out, and then look at what kind of expression triggers it might be having.
Okay. Thank you very much. It's he is in time, so thank you very much for his nice talk. Okay, Sharmila, please.
Thank you, Subhrata. And at the outset, I would like to extend my sincere thanks to Orunda and Ashimadi for giving me an opportunity to be a part of this program. So the next speaker of this session is Dr. Preetha Ray. And Preetha is a scientific officer G and a professor in translational research. Preetha heads the Imaging Therapeutics Lab of ATREC Tata Memorial Center, Mumbai. She has a broad research interest. Mainly, her lab is trying to investigate and detect the key molecular switches associated with metastasis and chemoresistance in epithelial ovarian cancers as well as in gastric cancers. Besides, they're trying to develop new therapeutics and diagnostic strategies, and she uses a number of drug-resistant models in ovarian cancer and gastric cancer cells and patient samples to establish her findings. And she are using various kinds of emerging technologies. Her research has been funded by the Tata Medical Center, DBT, DST, CSIR, Terrifox, and LTMT. Preetha's educational background, she's again from Vishwabharati, as you have already heard from Murunda. She did a MSc from there and then moved to the Banaras Hindu University to do her PhD. Subsequently, she did her postdoctoral research in cell imaging at Stanford University at the UCLA. And she has received various Best Poster Awards and Travel Awards. Preetha has filed a number of patents, and both Indian and US, and she has published very well in high-profile journals. She's a member of various societies like the Indian Society of Cell Biology, Society of Biotechnology, and Indian Society for Cancer Research. And she is also, she has professional memberships of various renowned institutions and journals, etc. And with this, I would like to invite speaker Preetha to commence her talk. Thank you.
Okay, am I audible now? Yeah, thank you, Sharmila-di, for such a elaborate introduction, and good afternoon, all of you. To be very honest, before coming here, I have not really much aware of this society, and I felt really very good because this is a very unique society, I believe, where the legacy of a fantastic teacher, a mentor, is carried by excellent students who have done excellent in their own field. And the legacy continues, because when I got the call from Arunda and Ashimadi, I was really surprised and overwhelmed, because I have been fortunate to get taught by Arunda, not by Ashimadi, and looks like the teacher and student relation always continues, and I hope that will continue together. So today, as Ashimadi a little bit requested me that talk about my research, particularly on the cancer stem cell part. So before, as mentioned, I was in Stanford for a long time, and there the entire focus was on early detection of cancer. When I came back to India and joined Tata Memorial Center, I realized early detection is probably a myth in India, because there's a huge number of population and very poor medical health care. So, but my interest was always in early detection, so I little bit changed my course, and I would try to tell you what we are doing right now. There were excellent talk by Deepan and Shushantoda, who had already laid down the background of my talk. So cancer, as you know, today where we are standing, we have a lot of good treatment like hormone therapy, surgery. One minute. Sorry. So, yeah, the laser pointer. Oh, pointer option. Arrow is better, laser is not looking. Yeah, that's fine. So The hormone therapy, which is the base for the breast cancer treatment. Surgery is an ancient old century for cancer treatment, then BMT, chemotherapy, targeted therapy, and very recent one are the immunotherapy. And also there are a lot of advancement in the medical science, nursing, etc. So the patients are much more treated well, better than what they were getting the treatments 50 years back. However, the big challenge still now is that cure of cancer. The reason is that no matter whatever therapy is applied, these cells are so smart, they can change themselves, they can revert the receptors, the receptor switching, mutation happen, and they gradually become resistant towards like endocrine resistance, chemotherapy resistance, as well as the recent ones, the immune therapy. So the resistance is probably the biggest challenge for the cure for oncologists in 21st century. And as you all know, this resistance can be of two types. The tumor is a very heterogeneous population, so if we apply one drug, it may not affect all the cancer cells by virtue of their heterogeneity and their genetic constituents. So some cells probably would always be non-responsive to one of the drug, and they finally lead to the recurrence of the cancer. Whereas there is acquired resistance, the tumor cells being continuously stressed by the application of the drug, they start modifying their components by genetically or epigenetically, and finally give rise to the resistant cancer. So this cancer resistance can have, these tumor cells have multiple pathways where they alter, like genetic and epigenetic, the tumor microenvironment, the apoptosis, drug metabolism, etc. Over to that tumor cell, there is a very special group of cells which are cancer stem cells. I'm not going into detail because Sushanta already have told a lot about them. And these cancer stem cells is a group of population of cells. By virtue of the certain characters in them, they always remain untouchable by the drugs, and therefore they can lead to metastasis, they can lead to resistant tumors, etc. So, as I told, my lab is interested in two different types of cancer, epithelial ovarian cancer, where the prevalent subtype is the high-grade serous ovarian cancer. Another is gastric cancer. Luckily, India doesn't have a lot of incidence for both the cancers, but the major challenge is the late detection and recurrence due to the chemotherapy. So the survival rate for high-grade serous is less than 40%, 5-year survival rate, whereas for gastric cancer it's less than 20%. So today's talk I will be mostly focusing on the epithelial ovarian cancer, what we are doing right now. So this is just a brief snapshot of how epithelial ovarian cancer, particularly the high-grade serous ovarian cancer, are treated in the hospitals. So usually these patients come very advanced stage. They are given neoadjuvant chemotherapy, 2 to 3 cycles of platinum and Taxol, followed by surgery, another 2 to 3 cycles of, and they actually show very good response. Unfortunately, within few months, about 70% of them relapse. And this recurrent high-grade serous patients, they can be grouped into two categories. One is refractory or platinum resistance. These patients, the disease come back within 6 months of the last treatment, and they never ever show response to the platinum. We don't know why. And the other group, the recurrence usually 6 to 12 months or before, after, and they start showing the response to the platinum agent again. Again, we don't know why. So these are very intriguing biological questions which we tried to answer in our subsequent slide. And there are some approved targeted therapy for these particular cancer type. The VEGF, which is targeting the angiogenesis, the PARP inhibitors, which is applicable to only 15 to 20% of the patients, who have BRCA mutation, and very recently the FR alpha positive platinum resistant ovarian cancer, this marvetuximab is approved by the FDA. However, there is a large number of patients which do not have this BRCA mutation, which is around 40 to 50%. They do not have any chance to get targeted therapy. The reason is there that there is very few mutation present in this cancer except P53. The rest, all the signaling pathways like RB, PIK3CA or Notch, there are amplification of the genes that lead to the altered or signaling pathways. So even in the recurrent high-grade serous ovarian cancer, a lot of genomic studies have been done, and people did not find that when the tumor is coming back, there are targetable point mutations are being present. So the question remains, in this omic era, this molecular snapshot, is this enough to develop targeted strategies, particularly for recurrent high-grade serous ovarian cancer, where mutation rate is very low? So we tried to answer this question. So as I said that I'm much more interested in early. So when I started my lab, I asked the question, can we capture the event early when there is a development of resistance so that we come out some solution that will be beneficial to the patient? So we developed a lot of cellular model against cisplatin, paclitaxel both, and divided them early and late resistant cells. We characterize these very well, the cellular model. Intriguingly, we find that the early resistant cells, there is a high upregulation of insulin-like growth factor receptors, and this which goes down as the cells become more and more resistant. The activation of AKT going up as the cells gain more and more resistance. And the question is that now these cells are having, so what is the status of cancer stem cell population in these cellular models? So we looked at the cancer stem cell population by different strategies. One is using the biomarker, one is using the spheroid formation, another is using the inherent drug resistant property of these stem cells. Basically what we do, we incubate the cells with a DNA binding dye. The cells which have very high dye efflux transporters, they can actually throw out the dye from them, which is here captured as a violet little small percentage, and the rest of the dye cells have this DNA binding dye. So we capture this small population which are not fluorescence in nature, and what we have seen as for each of the model, there is increase in the CSC population or the site population as the resistance is developing. Cisplatin, paclitaxel, and combination model. And so this site population are the truly cancer-like stem cell enriched population. So we collected these population, cultured them, and we did the spheroid formation assay, and then you can see all the NSP population, they were not able to form spheroids, while the SP population were able to form spheroids generation after generation. Increased pluripotent gene expression compared to the NSP population. And then another assay what we did, we collected these cells, cultured them 2-3 weeks, again sorted them, and so when we collected, you can expect that this is the 100% of the cell. However, what we see, there is a enrichment in the SP fraction when in the second sort, which is from 5.8, and then more into 20% in 7.3, and in the dual positive cells, it's going around 20%. However, but the rest of the cells actually getting differentiated in the non-site population, which is a true characteristics of a stem cell. A stem cell can differentiate into a non-stem cell and give rise to the progeny. When we did the same assay by the NSP population, we did not find any more SP population for repeated culture of the NSP population. and the SP population are more drug resistant than the NSP population. So we could understand, yes, these SP fractions are enriched with the CSC-like population. We also implanted in the mouse, and these are the gold standard of the CSC population, that even if you inject 10 cancer stem cells, they are able to form tumors. So here we injected 5,000 of the SP cells and 50,000 of the NSP cells, and you can see that the NSP cells are not able to form tumor over time for both the models for the early as well as the late resistance cells. In case of the late resistance cells, we see that the tumor formation takes much longer time, indicating in the late resistance cells, this SP population is much more quiescent in nature, less proliferative than the early resistance cells. So all these lead to that these SP cells are enriched with CSC-like population. Now what I showed you that the IGF-1R expression is very high. You all know that the IGF-1R can act in two different arms, RAF-MAP kinase ARC pathway and the PI3K AKT pathway. So Aniket comes and he looked at all the activation of ARC and AKT and found that in the early resistance cells, there is a high level of activated ARC at the basal level, which increased upon the treatment of the drug, which doesn't happen with the AKT upregulation. This has been true for the downstream targets of the ARC. And in the early resistance cells, ARC is mostly localized in the nucleus than the late resistance cells. So what is the consequence of this high ARC activated ARC in the early resistance cells? He looked into many different mechanisms, and we narrowed down to autophagy. The reason is that autophagy, as you all know, it's a cell death mechanism, but it's actually for the cell survival mechanism. So therefore, we started looking at the autophagy. And for the students, the autophagy is basically, initially there is a small phagophore formation happens, then which complete by the phagophore and which fuse with the lysosome, and all the degraded proteins which happens due to different kind like starvation, other stresses, infection, chemotherapy, all the biomolecules get degraded, and these molecular units are being used by the cells for their own survival. And in literature suggests that ARC is involved in many steps in autophagy, and there are plenty of literature which are there to support this. So what we did, we inhibit ARC, and so, sorry, I could not zoom out, this option is not here. So inhibit ARC, and what we found when we treated the cells with drug, there is decrease in the P62, which is one of the adaptor protein in the autophagy for both the inhibitor, and when we use the, I should use the pointer. Pointer will not use gum, no? The laser pointer is not, option is not here. Anyway, so when we either inhibit autophagy or ARC, we did see accumulation of P62, which means that the autophagy is stalled when you inhibit ERK. However, when we use AKT inhibitor, the autophagy is ongoing. So there are two different mechanisms happens between these two kinases. ARC is highly activated at the early stage. You block ARC, there is no autophagy ongoing, and the cells are, I'm not showing the data, cells are dying. Whereas at the late stage, high AKT is present, and you inhibit AKT, autophagy is ongoing. So we also looked at in what step, basically, this ARC is playing role. So if you can really look in this little window, that there are a lot of yellow spots. So what we used antibody against LC3 and an antibody which will detect the lysosome. So wherever there is a fusion happens, you can see this yellow spot. Only in the early resistance cells, after the drug treatment, we did see a lot of yellow spot, which means the autophagosome and lysosomes are being fused, and therefore we believe that the ARC is playing a role in this particular step through UV-RAG and RAB7. So till date, in the field, there is a very little, 1 or 2 sensors are there, but in vivo, there is no sensor which can catch autophagy dynamics. So we built a sensor. The principle of the sensor is there is a thermostable firefly luciferase which is fused with the P62 proteins. If there is no autophagy, we see a lot of signal. If there is autophagy, there would be less signal. So this is a busy slide. A lot of mice are there, and we treated them. We implanted the tumors expressing this sensor, and we treated them autophagy inhibitor, ARC inhibitor, combination of drug and ARC inhibitor, combination of autophagy and drug. And I just want to show, just please look into this. When there is only drug, that means the autophagy is ongoing. The signal is always low. However, when we are using an ARC inhibitor or a autophagy inhibitor, there is accumulation of the luciferase because autophagy is blocked, and that accumulation only remains for 48 hours. So this particular sensor is so powerful that it can not only capture the dynamics of autophagy in vivo, it can also tell you how long the drug is acting, which is very important for any clinical application of the drug. But the question is that it's all happening in the early resistance cells. In the late resistance cells, we don't see the autophagy. Is it really true? So all the components of the late resistance cells, the CSC and the non-CSC, they have the similar or differential autophagy flux. So we basically looked into the different autophagy flux, isolating the SP, NSP, and the total population, and we find that in the SP population there is low autophagy, high autophagy, and in the NSP and the TP population, there are where the autophagy is stalled. The same thing we have captured by the microscopy as well as the transmission electron microscopy, where these electron-dense particles are actually showing you the autophagosome lysosome fused bodies, which are more in the SP population. So what is happening? Is there any molecular player which is driving this autophagy in this? We narrowed down to, after looking at many proteins, we narrowed down to the inhibitor of differentiation family, which is known for maintaining the self-renewal property in stem cells as well as cancer stem cells. These are basic helix group helic proteins which act as a repressor to the other transcription factor because they don't have any DNA binding domain. They interact with the other transcription factor and inhibit their function so that the cells remain quiescent in nature all the time, and that's the stem cell property. So what we find that in the SP population, the ID1 expression is maximum in both the cell lines, and when we induce autophagy, either using the taurine, which is the mTOR inhibitor chemically, or using serum starvation, the ID1 level goes down, and when we use the chloroquine or taurine plus chloroquine, the level stabilizes, which means that when there is a lot of autophagy going on, the ID1 gets degraded. What's the consequence? And which is the ID1 interactor partner? So by string analysis, we narrowed down to TCF4, TCF12, and TCF3. These 3 proteins, and finally TCF12. We did a lot of simulation study, and we find that out of the 4 ID family members, there are 4 ID proteins in human. ID1 has the maximum residues which can interact with TCF12 and the maximum strong binding looking at that negative energy. We also did co-immunoprecipitation with TCF12 antibody or ID antibody and showed biochemically that yes, these 2 proteins are physically interacting with each other from 2 different cell lines. Now, so ID1 is interacting with TCF12. and autophagy can modulate this ID1 degradation as well as the TCF12 expression. What is their link with the chemoresistance? So we looked at the different efflux protein, and we looked at their expression in the SP cells. We narrowed down to this HCTR protein or SLC31 protein, which is a copper transporter, also being used for platinum uptake by the cancer cells. So by CHIP, there are 6 TCF12 binding sites on the HCTR promoter. What we find that the SP cells, the TCF12 can bind much more on the HCTR promoter in site 1 and site 2 for both the cell lines. And when we use AGX51, which is an ID inhibitor, the level is basically much stronger in the SP cells, which means that when ID1 protein is degraded, the SLC31 or 31 or HCTR expression level goes high. What does it mean? What is the endpoint of it? If there is a lot of influx protein present, that means the cell is able to take platinum, lot of platinum there. So we did ICP-MS, which is the quantification of platinum inside the cell, and we did see that the SP cells are having low platinum than the NSP cells, and when you modulate with the ID1 is degraded, the platinum content in the cells are much more higher. So therefore, there is a complete axis, the ID1, TCF12, and HCTR axis, which is basically getting modulated by autophagy, and that determines the chemoresistance properties of the CSCs. Now, all this, what is the connection with the human? So what we did, we collected the tumor spheroids from different patients. Now, these patients, they develop a lot of peritoneal fluid, so it's easy from them to collect the peritoneal fluid and isolate the tumor spheroid. As I told you earlier, that the patient can be grouped based on their platinum response into platinum sensitive relapse or platinum resistance relapse. And we isolated these tumor spheroid and analyzed the autophagy rate, and you can really see that the platinum sensitive relapse, that the patients again show response to this, have very high lysosome and autophagosome fusion. And this is also, this is shown graphically. And when we looked at the HCTR or the copper transporter level, the treatment naive patients have low, and sensitive relapse patients have high HCTR level. The ID1 expression is just opposite. So this is something which we still were trying to understand, why some patients respond to platinum again in the relapse, and we believe that autophagy is the main modulator in these patients, and they basically make the patients sensitive to platinum through this ID1 TCF12 HCTR axis. So this is almost there, but can we use this axis as targeting therapy? So before having targeted, we need to understand what is the implication of this, all these inhibitors. So we started, this is the proposed model I already described to you, and we started looking at the transcriptomic profile of these AGHS patient-derived tumor cells treated with AGX51, which degrades ID1, and you can see that all the cell cycle-related and EMT-related genes are downregulated, which means the ID1 basically control these patients, and these two are chemo-naive, and this is the relapsed patient. This is just the stories is trying to beginning, and we are trying to look at different pathways and identify if we really use this particular inhibitor as a phase 1 clinical trial to the patients, what are the effects of would happen in the patient, and what are the pathways can be altered. So this story is continuing, and probably nothing can be done without this very enthusiastic group of students. These are my senior students who have already passed my lab and settled down in other areas, and this is my current ones. I have a very excellent group of clinicians, friends, and other scientists across other institutes, And of course, without funding, we can only dream but cannot do anything. So thank you very much. I will be happy to answer any question.
Thank you, Preetha, for your excellent presentation. This is now open for discussion. If there are questions. Yes, Shyamulda, please.
So, fascinating talk. So there are two things. Number one, that in early versus late, in early you get autophagy, and late you don't get it, and their growth kinetics is very different. And second question is that, well, how autophagy is basically related, because your flow cytometry, or not flow cytometry, the confocal data showed that the platinum resistant versus platinum sensitive, which is also linked with the autophagy. So theoretically, I would expect that the autophagy is a process. It could be double-edged sword. So in one way, it can take the cell for death, and other way for its rejuvenation type. So how do we explain this thing in the light of cancer cells?
Yeah. So a very good question. For answering your first question, what's the difference between early and late, and why in late we don't see autophagy? We know the answer. I didn't say it here. We see very high activated AKT. Now, activated AKT can also phosphorylate other transcription factor like FOXO3A, which, yes. Yes.
But biologically, what does it tell you in terms of tumor biology?
Yes. So the tumor is constantly experiencing stress. When it is going, it is experiencing a lot of stress from an genetic hypoxia, or even the drug itself. So what is happening, what we believe, that this set of patients, we are also doing the genomics of these patients as well. There are a set of patients where probably these autophagy genes are highly upregulated, and therefore they give these patients an advantage that when there is a lot of autophagy, the cells become more sensitive towards the platinum, because the ID is getting degraded, TCF12 is free, and it can bind with the HCTR, and then a lot of platinum can be uptaken by the cells. So the microenvironment of the tumor cells and is actually driving a kind of a force for initiating autophagy in these tumor cells, resulting them to be more susceptible towards the platinum. Whereas the platinum resistance relapse patient, which never respond to platinum again, probably that force is not there. So that is why this all transcriptomic analysis we have started, that why it is happening, and also the genomics of this.
Thank you.
Yes. Please. Yes.
So, it's excellent talk focused about cancer stem cell. So I have one question actually, but I mean, is there any way, or have you thought about, differentiating the cancer stem cell to a different type? And it will be something like terminal differentiation, right? So that they cannot again go back to the stem cells. So
Yeah.
is there any way, anybody has tried to do it this kind?
Yes. So targeting cancer stem cell is probably remaining the most challenge right now in the cancer biology. Number one, the population is very small at the beginning when the chemotherapy starts. Number two, it's very different because there is no specific biomarker. For certain cancers like breast, there are certain biomarkers have already identified, CD44, CD24. Most of the cancers, there is no biomarker. So how do you identify these cells and all? So one mechanism which I have elucidated here, that is leading to the differentiation of the cancer stem cell in a non-cancer stem cell, that is SP versus NSP. And this differentiation is based on majorly their chemoresistance property. So we can induce some certain condition to them. So one is that the autophagy. So the autophagy inhibitors are highly coming into the clinical trials for many cancers. What we have shown here, not the autophagy, probably more autophagy is leading to the platinum resistance. So identifying and targeting them is a challenge, but there are ways coming up that can make them differentiate in certain area where that can be tackled by the drugs.
That should be great. Actually, the stem cells can be differentiated, right?
Yes. So
But
that's
why not cancer stem cells? That's why.
yeah. So that's why I showed you that we see that the SP population, if we take the SP population, culture few weeks, again SP population, they get enriched, but not 100%. So that means they are differentiating into their NSP counterpart. Whereas when we are collecting only the NSP counterpart, they never can give rise to the SP population. So
Thank you.
Are there any questions from the students? Or if not, let's thank Preetha once again for her wonderful presentation.
thank you.
I would like to invite Dr. Shiv Shankar Ray for his presentation now. Dr. Shiv Shankar Roy is a chief scientist of CSIR IICB, and he's also professor of the Academy of Scientific and Innovative Research, CSIR, Indian Institute of Chemical Biology. As you've already heard, he has also done his MSc in Life Sciences from the Vishwabharati University and his PhD from Bose Institute. He went on to do his postdoctoral research at the Harvard School of Public Health, Boston. Some of his research interests is mainly focused
because this is a condition which is tumor cells are facing, and due to these conditions they are trying to adapt. Okay, so the stresses like nutrient deprivation, hypoxia, metabolic stress, immune surveillance, which is not stress but it's another factors, growth factor. So these are the different things they are facing, and tumor cells are also adapted like metabolic and bioenergetic adaptations, signaling alterations, structural modifications, and ECM remodeling, etc. and etc. So why they are adapting? Because of they are trying to survive and trying to propagate. So there are different attributes in the tumor cells. You know that this is already, this is very important and very widely circulated picture where you can see that different types of attributes in the tumor cells which is acquired by the tumor cells. So you are concerned about the metabolic alterations in the tumor cells, deregulating cellular energetics, and there are so many other avoid immune destruction and so on and so forth. So these are different changes occurring in the tumor cells, and these changes actually, when it is occurring in the tumor cells, researchers are trying to find out some sort of agent or factors that can block that particular pathway or something, so that you can check the tumor cells proliferation or different stages of tumor oncogenetic processes. So now our focus is on the tumor metabolic alterations, which are, tumor cells are always in the process of dynamic metabolic alterations in the tumor microenvironment. So I am just telling you that this is the different pathways and different modifications are being, tumor cells are facing or altering. So people are trying to find out the different pathways, altered pathways, and find out the different modulators to the tumor cell progression. So there are so many, the list is very long, and so one of the few lists has been enlisted here, where you can see that different modulators are targeting different pathways or different metabolic aspects. And I am just adding two things, like one is glutaminase and another one is like lactate carrier protein, hexokinase 2, because I will come into this aspect, why these modulators are in the different stages of clinical trial, but they may not work due to we find out something else. So, we are working in ovarian cancer. I am not going into the details of because Pritha has already talked about ovarian cancer, so I am not going into the details. So, why ovarian cancer we are working, and there are so many issues. Pritha has already mentioned one aspect. I can tell that the occurrence versus mortality rate compared to breast cancer, ovarian cancer is very high. So, the overall ovarian cancer occurrence is less, but occurrence versus death ratio is very high due to several reasons already mentioned. And so, initially we showed, this is a few years back, we showed that among different modifications in the metabolic aspects, we found that one very interesting modifications in the lipid metabolism pathways. We found that this normally it is found that the phosphatidic acids are present, phosphatidic acids, phosphatidylcholine, etc. But when it is a tumor cells, due to this expression of these enzymes, phospholipase A2 and autotaxin, these lysophosphatidic acids are produced in the tumor microenvironment in very huge amount, and these lysophosphatidic acids in turn acting as a signaling molecule in the for the metastasis of the ovarian cancer cells, and also it acts as a formation of unsaturated fatty acids inside the cells. So, growing proliferating cells need a lot of biomass, and also need the signaling pathways for the metastasis processes. So, we showed that this particular lysophosphatidic acid production is very high in the tumor cells, but compared to the normal cells, and this helps in the tumor metastasis processes. So, we showed that this is the signaling cascade. So, the lysophosphatidic acid actually overproduced, and it binds to the one G protein coupled receptor, which is also overexpressed in the tumor cells, and through signaling processes it induces certain oncogene, which in turn actually helps in the tumor aggressiveness by inducing the matrix metalloproteases and also metabolic alterations in the tumor cells. So, this is actually one of the example, and also you have recently shown that lactic acid is overproduced and goes to the nucleus, and this is actually promotes epigenetic alteration to induce the cancer cell proliferation. So, these are the some examples I am just showing because these are the modified metabolic processes which helps tumor cells to further proliferate or survive. So, so this is actually one of the review article we wrote to show that these different pathways, altered metabolic pathways can be, there are several can be targeted to check the proliferation of the tumor cells. So, this is one example. I am not going into the details because it is already published a few years back. So, we will show some sort of interesting information which I got recently, that is that nutrient deprivation, because tumor cells are always facing the nutrient deprivation. And the tumor is, there are a lot of blood circulation, and the core of the tumor will get less amount of nutrients. Now you consider the cells, the blood circulation where the glutamine is very high, it will be reached towards the surface, and in the core there will be less glutamine. So similarly, when there will be lot of glutamine, when there is a glutamine starvation, what will happen? That will be, similarly that will happen in the core of the tumor. So our question was, what is the effect of glutamine starvation on cancer cells, metabolism and growth? Is there any connection between the glutamine starvation and stemness and chemoresistance? Because the analogy is the core of the tumor is also having the more chemoresistant and the stem-like cells. And how to address this issue to get the better therapeutic strategy. So, glutaminase inhibition and at the same time using the Eldon, another chemical modifier which blocks the glutaminase, is the same. So, if you remove glutamine from the medium, is same as if we use the Eldon which blocks the glutaminase. So, now we did both without glutamine treatment as well as glutaminase inhibitor. So, probably this is almost similar phenotype will give. So, in the so I am just showing you the data very fast that stemness and chemoresistance properties are increased upon glutamine starvation in tumor cells. We showed that the stemness marker, that is we use the OCT4, SOX, NANOG and ABCG. So, these markers are highly expressed when you treat the cells, when you grew the cells in absence of glutamine, and also CD44, CD45 markers is very high in the glutamine starved cells. And without glutamine, the dry efflux population is higher and dry retained population is less, suggesting that dry efflux is more. So, now, so first we wanted to confirm that whether there is a sort of stemness acquiring is in these cells or not. So, this is actually showing some results, suggesting that stemness property might be there when you grow longer time with the glutamine free medium, which is predict, which you can assume, because the cells when is harsh condition, it might go to the induce some sort of stem property for its survival. So, now the next question is glutamine starvation. So, what is happening in the mitochondrial biology, mitochondrial structural and functional aspects of the mitochondria in absence of glutamine? So, the cells grew in absence of glutamine, and you can see there is a drastic change of the structure and localization of the mitochondria. You can see that this is the normal cell without glutamine, and this is with glutamine, and this is without glutamine. So, mitochondria are localized in the towards the nuclear region. This is called perinuclear localization. Size of the mitochondria is very short in absence of glutamine. So, this is the without glutamine, size of the average mitochondria is about 1 micrometer. And when there is this is the larger size, more than 4 micrometer mitochondria is found only in control without glutamine is less. So, size of the mitochondria is reduced and in absence of glutamine. At the same time, if you see the this is the line scan analysis, you can see the localization of the mitochondria in absence of glutamine is towards the nucleus. This is towards the nucleus and this is the normal. So, you can always see that this is going towards the nucleus. So, there are two things that are happening. Mitochondria is fragmented and it goes to the perinuclear region of the nucleus. So, this is the size. This is the video is not working probably. So, this is the size of mitochondria, is normal, and if you treat the cells with the glutamine, starve the cells with the glutamine, the size is becoming very less. So, now this is the mitochondrial dynamics. There is a fission and fusion always going on. This is the mitochondrial biology. You can so there is always a balance between fission and fusion, but the protein which is very important for the mitochondrial fission is the DRP1. So, DRP1 actually phosphorylated DRP1 binds here and it cleaves the mitochondria. So, DRP1 is the important protein for the fission. So, our interest was whether glutamine starvation induces a DRP1 phosphorylation or DRP1 expression or not. So, so to find out about that, we found out that DRP1, phospho-DRP1 is very high in absence of glutamine, and its localization and other things, and co-localization also suggested that it is highly expressed and it is localized in the nuclear. It is highly expressed, and also we showed that without glutamine, this is without glutamine, and this is the without glutamine and without DRP siRNA of DRP1. You can see the difference. This is without glutamine, scrambled siRNA of DRP. This is with DRP siRNA. So if we block the DRP1 expression, so this is actually not happening. So DRP1 is playing the major role in mitochondrial fragmentation in absence of glutamine. So absence of glutamine actually induces probably the DRP1 phosphorylation, which cleaves the mitochondria in this condition. So next we checked that DRP1 whether is playing the role of stem-like structure or not, and we found that with the FACS analysis we can find that this is scrambled DRP1, this is the siRNA of DRP1 glutaminase. You can see that is a stem-like features is reduced, and also the efflux population and drug retaining population you can compare, and we showed that yes, DRP1 probably is the important factor which is playing the role in absence of glutamine. So far we showed that this DRP1 phosphorylation at 5S616 position actually helps in mitochondrial fragmentation. It reaches to the perinuclear region and something is happening. What is something is happening? We don't know. We found out a couple of papers and maybe another few papers suggesting that perinuclear localization of the mitochondria, nuclear localization of the mitochondria helps create some sort of environment, localized environment. It helps in the expression of certain genes. They have already shown that perinuclear mitochondrial clustering creates an oxygen-rich nuclear domain required for hypoxia-induced transcription. So it is possible that it does something where in the expression of the stem-like genes like OCT4 nano SOX, these genes are highly expressed in glutamine-free condition. So we don't know about it, but we are trying to find out what is the mechanism. So, but what we want to do is, so glutaminase inhibitor is already in the clinical trial, and it is actually very good drug for the tumor reduction. It is naturally tumor will regress. If you treat the animal model, large tumor, glutaminase inhibitor if you put, tumor will regress, and it will be very effective agent for the tumor regression. But our study suggests that this glutaminase inhibitor which is in the clinical trial might not be useful because it might create some sort of stem cells, cancer stem cells, and it will recur. So our study suggests that it might recur because of the stem cells. Now we wanted to use that both DRP1 inhibitor and glutaminase inhibitor as a combination to see whether they can reduce the growth as well as they can actually take care of the stemness property so that the stemness cannot occur in the tumor cells. So we tried to do that in an animal model, and you can see that this is the L-DON, which is a glutaminase inhibitor, which when we treated, tumor regression occur, but this marker ABCG2 and OCT4 SOX2, these are actually expressed. But when you treat the animal with the MDV1, which is a DRP1 inhibitor, and the L-DON glutaminase inhibitor, co-treatment actually regress the tumor, as well as you cannot see the stemness marker in the tumors. So it suggests that if you just co-treat with the tumor regression, means L-DON to regress the tumor, and also the MDB1 DRP1 inhibitor for the stemness reduction, it can be a good drug. So this is actually our this is hypothesis, and Parosh and Sampurna actually did this work, and they published. And so that's why I'm telling this is one CB839 glutaminase inhibitor, which is maybe in the advanced level of the clinical trial for the different cancer types. It might not be good because it might create some sort of stemness recurrent will come. So means this type of metabolic alterations and metabolic inhibitor studies can give some idea, so that we can use the combination to address the issue in a bigger way. So this is one example. So in another aspects, we can tell you that it is very recently we showed that this DRP1 in absence of glutamine, so this DRP1 actually induces a stemness, and sort of in absence of glutamine, stemness occurring. But in presence of glutamine, actually through some oncogene, ETS1, the DRP1 phosphorylation induces the oxidative phosphorylation and glycolysis, and it induces the EMT and invasion. So this is maybe context specific. In absence of glutamine, DRP1 is doing the fragmentation of DNA going to the perinuclear region and doing some jobs. In presence of glutamine, when there is no need of the stemness property, this DRP1 fragments the DNA, but it doesn't go to the perinuclear region. It actually helps in oxidative phosphorylation, enhances the EMT and invasion through metabolic alterations. So it is possible that same protein might function different way when there is different contexts. So this is actually we have recently shown. There are plenty of another example. I am skipping, but the time may not be there. But where we have shown recently, it is a couple of days back. It is published accepted in BBM also. So where Deepshika and Preeti has shown similar way, they have shown that what is that? MCT, this is lactate transporter, as well as the hexokinase, when they use the inhibitor of both, it is actually taking care of two functions, that is lactate mediated proliferation and also the glycolytic inhibition. So this is a long story, but there is no time today. But this is another example of using the metabolic inhibitors, two metabolic inhibitors to block the tumor cell population. So they have used two drugs, that is 2DG and alpha CHCA, to try with this, and they found a very, very interesting result, which supports that dual inhibitor can be a good way. So that's why I told that single inhibitors, they are already in the market, but there might be better options are there if you use the two target oriented inhibitors. So finally, my conclusion is the metabolic pathway modulators might be promising candidate for the tumor cancer therapy, because tumor cells actually are modified, as I mentioned already, and these modifications can be exploited by using different modifiers or inhibitors to block the pathways. So this is all about today. Thank you very much. Actually, and this main work has been done by Parosh, Sampurna, Preeti, and Deepshika, and I thank all my collaborators and other lab mates. Thank you very much.
Thank you, Shibu, in your right time. So, any question, please? Okay, go ahead.
So against the backdrop of your extensive work, So can you throw some light on the potential connection between, metabolic syndrome like type 2, obesity, and cancer?
So actually, I am my interest always in the metabolic functions. So I use cancers as a maybe model or something. So so there is a huge connection of metabolic diseases and cancer, because cancer itself is a metabolic disease. Okay. So I have already mentioned that out of 10 attributes, one is the metabolic. In a very first slide I showed. This is so this is actually, you can see that deregulating cellular energetics, which means the metabolic alterations are there in the tumor cells, which is attributes. It must be there. And so cancer is a metabolic disorder, there is no doubt about it. Now the question is other metabolic diseases, mainly obesity, diabetes, and this type of diseases, whether they are linked or not. So I don't know about the direct link, but obesity is the central to many diseases, many metabolic diseases. So obesity has a lot of link with other diseases, directly or indirectly. That I can tell, but whether diabetes is associated with cancer, that type of very simplified statement I cannot do. But obesity is the central to the all metabolic diseases. So it might have connection with many other diseases like cancer. So,
thank you.
Shamalda, apni question korei jacchen. There is no prize for you.
Shivu, hey, stop. So your mitochondrial degradation, and your stem cell, cancer stem cell, they are metabolically quiescent, or because the mitochondrial function is probably disturbed in your cancer stem cell because of the mitochondrial degradation.
No, I didn't show that data. Mitochondrial load, ATP production, and other mitochondrial function is not affected. Mitochondrial load is not affected, ATP production is not affected, so this is not affected. What it does, probably it is actually fragmentation occurs,
Right.
but total mitochondrial load is not changing. What we didn't show, but in the paper it is there. But what it does is, when there is a some sort of deprivation or stress or something, this mitochondrial fragmentation followed by its localization is very, very important. So so far I presume, because when there is a invasion occurring, mitochondria is localized to the apical region of the tumor cells. They are giving sort of energy with interaction with the actin cytoskeleton. So it is possible that this localization of the mitochondria followed by its function, which is not known probably.
But ATP you are getting, it could be Warburg effect.
This is different, but this mitochondria related to ATP, we didn't find much change.
So it could be Warburg effect, and therefore you are getting quantum of ATP production as same.
This is different. ATP production, glycolysis is mitochondrial free, mitochondria independent pathway, right? So whatever the glycolytic ATP is produced is a mitochondria independent. So, right, so ATP production, I didn't find any change. We didn't find any change. So what we find is DRP induced mitochondrial fragmentation is increased, mitochondrial localization is altered. Towards the nucleus. So that much we know. We are getting some supporting data of 3-4 papers where they show that some sort of oxidant-rich gradient or something is happening in the nuclear region that might create some sort of transcriptional alterations of certain genes. That is actually, it is very gray area. I don't know.
Okay, thank you very much. It's a nice session. thanks to,
আমি একটা প্রাইজ পাওয়ার জন্য বলছি। No, these things, the comment that he made, as also the answers that the questions that Samuel asked, reminds me of one book by Siddhartha Mukherjee, in which the introductory part was this, which I think the president mentioned in our talk to start with, that it's really a complex disease. It's not a disease, it's a combination of many diseases. And whatever we find in all the effects of the different metabolic events, like the one just Shamul asked about the mitochondrial dysfunction or whatever, all these things are found in a number of other places in fragmented way, in different systems. You look at, you mentioned about this kind of stress, you identified cancer as a stress. Even if you find that you will have different aspects. And to me, it comes out as a conclusion that it's always like that your 6 blind persons went to see an elephant. And cancer biology, is it true that cancer biologists have gone to in different ways and found out, well, this is the leg, this is the tail, this is the ear, something like that. All put together, is it true that cancer disease is actually combination of many things put together? It's a physiological disbalance
Yes,
somewhere.
absolutely.
Biologically, it's a different thing which people see at different ways. The different genes that all those speakers talked about, like Pritha mentioned a number of genes, transcriptome data, or the RNA sequencing data that he showed, all these things put together, to me, that this is something, so many things combined together, and it's only a snapshot that each
Yes,
and everybody
absolutely.
is looking into. Nobody is right, nobody is wrong. All put together, this is the picture. Am I dumb?
No, this is true for many
I
other
got
diseases.
everything.
Many other diseases. So snapshots, one snapshot, and side by side another snapshot makes the
Yes,
full
everything
photograph.
is important. So everybody is right, but nobody is 100% correct.
true.
Nobody is truly correct.
No. Every big snapshot, small snapshot is also a story. So
Yes.
right.
That's true.
But this is not the complete story.
So you cannot do many things in one life, right? So you are trying to find out something. Right.
Arunda, thank you very much. You are probably right. It's a combination of something. So hopefully in the next 50 years we'll be doing the same kind of research.
Fortunately,
in
we don't have
different ways actually in
Fortunately,
cancer.
we will never get the answer in our lifetime. Thank you very much.
So thank you to the 3 speakers, Arindam, Pritha and Shivu, for your nice talk. What to say? Nothing. So we are done for this session. Thank you very much.
It's a great pleasure for us to felicitate the speakers, the invited speakers. And I request Dr. Oshiva Lahiri Mojumdar to first felicitate Arindam. One of the chairpersons, please. Come help her. Shubhu to uthaye na. Tu uthaye na. ওকে না দিলেও উঠবে না কে ওঠে অত Second, may I request Pritha? Pritha ki hai? আমি তো মাইক্রোফোন ধরে আছি রে এই যে This is the two Shantiniketan students together. Three. May I request, please, Harishu, look over there, please. Shushanto, please. Shushanto Debe, okay. To felicitate one of the chairpersons. তোমার ব্যাপার কাকে আগে দেবে Ladies first, okay. Thank you very much, and thanks to the chairpersons as well as the speakers. We straightway go to the next session, which will be presided over by Professor Mala Mitro, and Shomshubro will conduct the session.
Good evening. Good evening. Good evening, everybody. We have now come to the concluding session. So we started with good morning, then good afternoon, and now good evening. This session, in this concluding session, will be presided by Professor Mala Mitro, the Dean, School of Science, Sister Nivedita University. Professor Mitro has done her PhD on the theoretical cosmic ray propagation from Indian Association for the Cultivation of Science, Kolkata. After that, she completed her postdoctoral research on cosmic ray from Indian Association for the Cultivation of Science, as well as she has another postdoctoral degree, postdoctoral research, which on neutrino detection from the Department of Physics and Astronomy, University of British Columbia, Vancouver, Canada. With this brief introduction, I want to request Professor Mala Mitro to take the stage. And I want to also request our mentor, Arun Lahiri Mojumdar sir, to kindly felicitate Professor Mala Mitro.
Thank you, Shomshubhro. And I'd like this opportunity to thanks to the organizing committee of Central Calcutta Society for Advancement of Human Development and Research. And it was, I couldn't be here because of various assignments, but it was indeed extremely organized and extremely, extremely enriching session, which I know that our students and all the faculty members and all the dignitaries who took part in this conference enjoyed very much. So thanks to all, especially the organizing committee and our Department of Microbiology and Biotechnology to help for this organizing this kind of seminar. And special thanks goes to Professor Arun Lahiri Mojumdar, definitely. So I'd like to call up on stage the students who basically, the first one, I'd like to request the team from Department of Life Sciences, Presidency University, Niranjan Ghoshal, Dibisha Biswas, and Ranjana Pal. Their poster is on TRAIL induces breast cancer cell proliferation via decoy receptor 2 mediated upregulation of cyclin D1. So if they are, someone, okay, you are representing the team. Okay, please come.
Yes, ma'am. Hello. So may I start? Yeah, okay. Okay, yeah. I will complete in that. Yeah. So good afternoon, everyone. Myself, Niranjan Ghoshal from the Presidency University. And the title of my talk is TRAIL induces breast cancer cell proliferation via decoy receptor 2 mediated upregulation of cyclin D1. So what is TRAIL? TRAIL is basically a TNF, tumor necrosis factor associated death-inducing ligand. This can be a very good choice for an alternative chemotherapeutic drug. And if we look at the TRAIL signaling, TRAIL basically acts through DR4 and DR5, and when TRAIL binds to the DR4 and DR5, it induces apoptosis. But several cancers, along with breast cancer, show TRAIL resistance because there are two other receptors called decoy receptors. When TRAIL binds to the DCR2 or decoy receptor 2, that inhibits TRAIL-mediated apoptosis. But this TRAIL and decoy receptor signaling pathway is not well elucidated. So in this study, we aimed to look at what happens when TRAIL binds to this DCR2 receptor. And the overall aim was, what is the role of DCR2 in generation of TRAIL resistance? So these are the experimental group. We are using MDA MB231 cell line, and it is also validated, the work is also validated in MCF7 cells also. So the experimental groups involves control cell, DCR2 overexpressing cell, control plus TRAIL treated cell, as well as DCR2 overexpressed plus TRAIL treated. And we have done using this control and DCR2 overexpressed cell, the RNA sequencing analysis, and we have obtained that when trail is binding with DCR2 overexpressed cell, the major pathway that is being upregulated is G1 S phase transition. And from the cytoscape analysis and hub gene analysis, we have found out that cyclin D1 was the most important hub gene that are being overexpressed when trail treatment was done in DCR2 overexpressing group. We have also validated the data in transcript level as well as protein level. And we have also obtained the information that when merely DCR2 overexpression is not causing the proliferation, when trail treatment was done over the DCR2 overexpressing group, the cyclin D1 localization is increased. This also reflected into BRDU cell proliferation assay as well as the flow cytometric cell cycle analysis, where you can see that the DCR2 plus trail treated group, there is a very increased peak in the G1 S phase proliferation, G1 S phase transition, I may say. And also in the BRDU plus BRDU assay, we can see there is an increased uptake of BRDU when trail treatment was done over DCR2 overexpressing group. So, cutting the long story into short, when basically this is a strategy that is adopted by the cancer cell to overexpressing DCR2 spontaneously. And when DCR2 overexpression was done and trail treatment was given, the more cyclin D1 is localizing into the nucleus, and that resulting in the G1 S phase transition. So basically, trail, that is basically a apoptosis inducing ligand, but cancer cell can repurpose that ligand to utilize it for its own proliferation. So what is the significance? Like, this study for the first time is saying that the TNF associated ligand can also act as a proliferating agent. And for the future plan, we are planning, first of all, this is a mechanistic approach to elucidate the trail resistance, overall trail resistance. Not only that, we have obtained several receptor as well as several molecular markers that we can block, so that we can evade or we can bypass the trail resistance. So, in acknowledgement, I would like to acknowledge the Central Calcutta Society as well as SNU for giving me this opportunity, giving our team this opportunity to present my work, my PI, Dr. Anjana Pal, all the lab members of Presidency University. Thank you.
So, thank you. You have covered in right time. So the next will be from Shoindhobi Ghosh from Department of Life Science, Presidency University, Kolkata, and you are also attached to Department of Biological Sciences, ISAR, Kolkata. So your time also 5 minutes.
Good evening. I am Shoindhobi. My title of my work is Analysis of Alternative Splicing Events in Autophagy-related ATG7 gene in context to decoy receptor-mediated trail resistance in breast cancer. So, as Nirajanda has told about the trail and its receptor story, I would like to add 2 more things with in the background story. First one is, like, you know about trail and its receptor. Trail is an TNF group of cytokine, and when it binds with DR4 and DR5, it induces apoptosis in cell, and in turn, when it binds with DCR2, it doesn't induce apoptosis, rather it induces some cell proliferating signaling pathway. Another aspect is autophagy. It is also related to cancer cells. As we all know, in normal cell, autophagy is a tumor suppressing mechanism, but in a stress condition like hypoxia and other stress, the cell can use autophagy as their escaping mechanism. ATG7 is an E1-like ubiquitin-like protein, which helps in elongation of phagophore and finally forms autophagosome. Another aspect is alternative splicing. As we all know that pre-mRNA splicing is a very important post-transcriptional event. In alternative splicing, exons are removed and exons are joined back together and introns are removed, but this happens in some alternative way, like the exons are joined back together in different combinations, thus different proteins are forming. So my workflow is basically finding the sequences of proteins, prediction of structures, validated the structure, and from there functionality assessment. So we have first we have used the same RNA sequencing data. There are two groups. Group 1 is the trail-treated MDAMB231 cell, whereas group 2 is the trail-treated cell with overexpression of DCR2. This is a sashimi plot we have got from the RNA sequencing data. The peaks are denoting the read densities or expression values of exons, whereas the arc denotes raw number of junction read. So what we get from the data, we got that for the control group or group 1, there is the whole ATG7 protein is expressed, whereas a skipped exon event is happened for the group 2, like DCR2 plus trail treated. We have predicted and refined the structures of both the isoforms, the longer isoforms and the shorter isoforms. We have validated the result using Ramachandran plot. And then we have done a sequence-based functionality prediction, which is similar to our structure prediction also. What we have found that for the whole, like the longer isoform, both the N-terminal domain and the C-terminal domain retained, whereas for the shorter isoform, only the C-terminal domain is retained completely and the N-terminal domain is skipped. So from this, we have concluded that a loss in this N-terminal domain lead to a loss of the phagophore elongation and the autophagosome formation of ATG7 in the shorter isoform, whereas the C-terminal domain, as it retains, in both the isoforms, the homodimer formation will be retained. So with this, also like it has a correlation with the previous works of Nirajanda, where we have found that trail is basically showing when it is bind with DCR2, it is basically showing a cell proliferation signaling pathway by reducing autophagy. So with this, I would like to conclude my talk, and I would like to acknowledge my supervisor, Dr. Ranjana Pal, and Department of Presidency University, and all of my lab mates and SNU also. Thank you.
Thank you. The next from Department of Biotechnology, Sister Nivedita University, I'd like to call up on stage two students are there, Oindila Acharya and Shomita Reja. So Oindila is going to present.
So good afternoon, everyone. I'm Oindrila Acharya from the Department of Biotechnology, Sister Nivedita University. So today I'm going to present about my talk. The title is A Cheap, Eco-Friendly Antimicrobial Solution for the Hospitals by only using lime and chlorophyll. We know we use bleaching powders and sodium hypochlorite as a bleaching agent or antimicrobial agent in hospital. But it causes chlorine toxicity like eye redness, rashes, and etc. So why not we look for another substitution? So that's how we got to the idea of using some herbal contents like only the chlorophyll extract from any plant, like we have used spinach here, and the lime extract, which is easy to available and also eco-friendly. So here is the experimental method. So we went to make some composite using this lime and the extracted chlorophyll from elly leaf. After that, we went to the muffle furnace for 180 degrees centigrade for overnight. And then we gave it to the light radiation to activate that chlorophyll. As we know, chlorophyll is a light-sensitive compound. And after that, we have performed the antibacterial activity, which is our aim. So here we can see, at first we have gone through the solid MIC results. So in the controls, there is no zone of inhibition, it's 0. But as we have used our sample gradually in different concentrations, we are seeing there is a large zone of diameter. Large diameter is also a zone of inhibition here, which is a great significant result of an antibacterial activity. And we have also used it in 5 pathogenic bacteria. Then again, we have proceeded to the liquid MIC results, where you can see in normal culture plus nutrient, the bacterial OD is very high. But as we have used our sample gradually, then we can see the OD is decreasing significantly, which is near to 0 even. So that's how we proceeded. Then we have gone to, after this in vitro, we have gone to the in vivo, because after prokaryotic cell line, it can be more significant if we can use that in eukaryotic cell line too. So we have gone to the animal models. And after this animal model, we have used in mice model, where the wound has been created after making that mice unconscious. Then we can see, we have observed it for till day 12. If I have kept that wounded mice for day 12, just normally, then we can see the wound is still here. But as we have treated with various concentration using that sample, making a bandage, we can see the wound is getting like healed in the day of 12th. So we can also see the percentage of that wound diameter, which is decreasing gradually and significantly. So we have proceeded further with this experiment. Now, we can say that the hypothesis is that after reading through a lot of research papers, that it is actually inducing singlet oxygen or ROS, because the chlorophyll present in this is inducing plasmon effect. This plasmon is getting into any biomolecule, and then it is making ROS, which is hazardous, and killing the bacteria. Now we are proceeding to make the quantification of the ROS. Now we are doing HPLC, LCMAs to prove our result, because we need to make it an industrial project gradually. Because it's a cheap, eco-friendly, sustainable. We don't need anything so expensive. And there is no toxicness. It is eco-friendly. And it can also be converted to powder form and also as an aerosol form, which can we spray in the air. So it can be a large-scale industrial business too, if we can proceed further with proper research and proper experimented. So at last, I would like to control, let's do something eco-friendly, cheap, and sustainable for this new world. At last, I'd like to acknowledge Dr. Somshubha Thakur Chaudhuri, my mentor of Department of Biotechnology, Dr. Ramananjan Bhattacharya, Shomita Reja, my co-worker, Dr. Saptoshi Das, and also the Department of Biotechnology and Microbiology, our Assistant Editor, Dr. Rajatpal. Thank you.
Thank you, Windila. So we have heard the students. These are the 3 best among all the posters which we could see. So the first one is first presentation, that is trail induces breast cancer cell proliferation via decoy receptor 2 mediated upregulation of cyclin D1, presented by Niranjan Ghoshal. So I'd like to congratulate Niranjan and his team, of course. And the other 2 presentations almost, I can say that they have done a tie. Still, if we have to choose among them, then the second one is from the same team, Niranjan Ghoshal, Shoindhobi Ghosh, which has been presented by Shoindhobi. So the team of Presidency University and ISER Kolkata, analysis of alternative, the second one, basically analysis of alternative splicing events in ATG7 gene in context to decoy receptor mediated trail resistance in breast cancer. So clap for Shoindhobi, Niranjan and their team. And that very close third is the team just presented by Oindila Acharya, Shomita Reja, and their mentor, Shomshubra Thakur Chaudhuri, Ramaranjan Bhattacharya, and Saptoshi Das. So they are just presented, so I'm not going to repeat the title anymore. So congratulations to all the winners. So I'd like to hand over the certificates to them.
I would like to call the students who have owned a prize in poster presentation. First, Niranjan Ghoshal. Niranjan Ghoshal. I would like to request both Mala ma'am and Arun sir. Simple solution. Niranjan Ghoshal. Now the second prize in poster presentation, Shoindhobi Ghosh and her teammate Chandan Das. Shoindhobi Ghosh and Chandan Das. Now the third position in the poster presentation, Wendrila Acharya and Shomita Reja. Thank you, madam. With this, we are concluding this session as well as our one-day national symposium. Now, the last part, that is the distribution of certificates. Many of the students are waiting, I know. Now the certificates will be distributed from the outside where the registration will take place in the morning. I want to thank all of you for this beautiful evening, beautiful day, the whole day symposium. Thank you, everybody.